Now we move to the next speaker. I don't need the paper. It's Professor Satya Prakash. He came from India, and he got his bachelor and master degree, and he's very good in fermentation. When he came to me for PhD, he came with an Indian international scholarship. I didn't know what it meant, and I found out later that it's supposedly one for each specialty for the whole India. Considering how many billions of people in India, it must be very difficult. So he did a very good job when he came here, and I gave him 2 projects. Always do that for graduate students. One is that if you get it, you'll be very good. The other one is that if you don't get it, you do another project and you can finish with a master degree. So he finished an impossible project, encapsulation of genetic engineered cell, and showed that it worked in rat to lower the urea level. And also later, we decided that FDA wouldn't like you to take anything genetic engineered. So we look at lactobacillus, which is used in yogurt. And again, when he finished his PhD, I said, that's your project, it's up to you. So he went and extended, and now he did so well, he has a company, and he's director of the company, and he's continuing to develop other area. Professor Satya Prakash.
Okay, good morning, everybody. First of all, on behalf of Artificial Cells, I would like to welcome you, all of you, as a family. And I would like to give you 2 experiences of mine when I started working with Dr. Chang. The first thing, when I came from India, he mentioned one thing, that not to be too shy. I was very shy, and that was the first lesson I learned. So I want everybody who has a red color badge, raise your hand and clap 3 times to thank him for all his. He actually made us complete people. So I really want to thank you. The next thing I want to thank him, I met my wife here in Montreal, and my father and mother, they could not come to Montreal. So Dr. Chang and Madam Chang, he represented my mother and father. So this is a story that he not only cared about us academically, but he cared about us personally so much. Thank you, Dr. Chang, again. So, as a father, you always try to copy that, and in that sense. But first thing what you do, you don't want to do what he did. As a good kid, you try to go away. So he did all his work that could have been resulted into billion-dollar industry, in my knowledge. So I said, okay, he did not do any of this, and so I said, let's do something that what he did not do. So I started a couple of companies. So I'm the co-founder of these companies, and he did not commercialize his work, but I tried to do that in a sense that let's do this way and let's see if I can make an impact. So I'm the co-founder of these 3 companies I want to disclose. And all the results I'm going to present today, they are not done at any of the companies. So it's university side that I have done the work that I'm going to present to you. So, other thing I follow him is to use artificial cells to do many kind of applications. So this is the lab that I work, Biomedical Technology and Cell Therapy Research Laboratory. And we do artificial cells, microbiome, cell therapy, biomaterials, drug delivery, nanomedicine, and biomedical devices. All these areas based on artificial cells. We touch upon all those. So today I will be discussing about microbiome and some medical devices. Microbiome I touch because human microbiome research has been really in forefront for the last 10 years, 15 years. And as Dr. Chang was saying that I did my PhD with him, and that time we proposed to use a genetically engineered E. coli to treat kidney diseases, to remove urea and ammonia. And at that time, we proposed obviously it's crazy idea to feed people E. coli, not only E. coli, genetically engineered. And that time, nobody actually had thought about it. So we started doing that. At that time, there was no term microbiome. But he was visionary to give me and that project and the challenge, and we did it, and we published in Nature Medicine. And that was one of the key publications that started, I think, an area of microbiome that, yes, one can use one bacteria to treat a particular disease. So I will talk about introduction about microbiome a little bit, about importance of microbiome. I will introduce your gut microbiome, and I will introduce your term probiotics and how one can use in heart health and some limitations and conclude with that. So let's look at the microbiome. So what is a microbiome? Well, everything inside us. Not only that, around us. And as a community in this room, we are being exposed to various bacterial cells, helping each other, transforming each other, infecting each other. This is our microbiome. So all this is affecting our human health. In fact, economics, they published that not only affecting human health, but affecting our metabolic pathways. And perhaps we don't know how. And till today, I think there are many diseases, including obesity and all kinds of diseases, where this has been known to be affected by microbiome. So gut microbiome is the area where the most we are being affected. If you can imagine that we have a huge number of bacterial cells in our body, and particularly in the GI tract, we have a huge numbers. And all these are affecting us every day, in fact, every minute, and now it has been proven to affecting our mood to everything that we can think of. Here I would like to introduce you two things. Number one, good bacteria and the bad bacteria. Bad bacteria you all know, and perhaps good bacteria also you know. We call them probiotics. So the idea is when you have a good bacteria, you are healthy, and if you have a bad bacteria, you are sick, particularly in GI tracts and everywhere. So the idea is to bring the beneficial bacteria from outside to make you healthy. Can we? So this is where I will give you an example of in heart health. Yes, we can. Heart health, particularly think about cholesterol. Cholesterol is a molecule that has been implicated in the heart health. And heart health, in fact, really is very dangerous. Almost 23 people worldwide are being killed. And cholesterol is one of the major risk factors of that heart health. And you have a high cholesterol, then you have a greater risk of all these diseases. So we targeted, and this is the numbers that you have a total cholesterol. And the US is 320 million prescriptions per year, about $26 billion market, just US itself. That's a huge number. And it looks like these are the guidelines. Every year the guidelines cover more people. So you got to manage this one way or another. So people are managing it with many ways. And let's look at the cholesterol, how it is, what is important in this is that it comes from diet 15%, but the rest in body is synthesized in our body itself, 85%. Therefore, we can manage through this, but this is where the main problems is. And why we have a cholesterol? Yes, this is because we have a lot of good stuff coming out of cholesterol, from our brain structure to hormones to almost everything. So I would like you to focus on bile salts. This is also a byproduct around the cholesterol. But before going that, think about atherosclerosis. We have a lipid deposit in your arteries, and as a result, you have a heart attack. So this is one simple thing where the cholesterol plays a big role. If you have a more cholesterol, you have a more deposit. If you have a more deposit, you have a more chance of having diseases like this atherosclerosis. So there are many drugs available to treat cholesterol, including statins, all these chemical drugs, and people also try to do dietary changes. However, even though they are very effective, still there are 50% people in the world cannot get it because even they qualify because this is a cost and expensive and all those things. As a result, there is a need to develop something not expensive, All these drugs, most of them, they work on these pathways, one way or another, through either liver metabolism or through pathways of bile salt hydrolysis. So what we thought, maybe it's a good to look into these pathways and see if any bacteria can manipulate that pathways. And this is, it looks like that how expensive it is. FDA just says, 14,000 per year, and $120 billion by 2030 is going to be that cardiovascular disease prescription and cholesterol-related diseases. So we thought, let's look at these pathways and see if we can manipulate this. So imagine you have a bile salt here, and if you have a bacteria that can somehow degrade this bile salt into different products, then potentially what you are doing is changing the whole pathways. As a result, you might see the influence of these bacterial salts into the cholesterol pathways. And this is what I started working on after finishing my PhD with Dr. Chang, from genetically engineered to a lactobacillus natural products and naturally isolated. So what we started doing is we started selecting bacteria. So we started with 350 strains, and we qualify them, disqualify them, discard them based on all the steps. We found that 3 or 4 are really good. And we qualify or disqualify them based on their bile salt degradation capacity, their stability, and their usability in food or drug or any other formulation. We don't want to get into the genetically engineer anywhere that is disease causing. So based on in vitros, and we analyze their genetics, and we try to see that if there is any manipulation in that genetics can help us. We also try to figure it out that how similar they are in terms of our own genetic metabolic profile of the bacterial cell that we have in our body. So we come up with all those things, and we found, and by the way, we named that bacteria as a cardioviva. So we tested this in a human GI model that we have developed at McGill. There are only few in the world. If you like to see anytime and you want to collaborate, you're always welcome to use. We have it. So what we does is this model is essentially a fermentation of bacterial salts, and in these vessels, what we grow is human volunteer fecal samples. And within 3 or 4 months, we get stabilized all the bacterial cells that what we have in our GI tract. And once we feed these cells through a computer-controlled process, we could see the manipulation of any bacterial flora anywhere it's happening. So this model is available for anybody. So using this, we started doing the work for cholesterol. And then what we did, we had hamsters. We feed them coconut diet. Coconut is very high rich diet, and please don't eat it because really you have a high cholesterol, and unless you have a special kind of genes. So on these hamsters, we feed them coconut, and we started giving the artificial cells containing this much bacteria, and we monitor their cholesterol level. What we found that a decrease in 29% and total cholesterol LDL 37% and all kind of changes. So we see that in animal models, these bacteria, given a single dose, have an impact. We brought that to the human, and we collaborated with Richardson Center in Winnipeg. This is a center for functional food and nutraceuticals. So what we did is we had artificial cells containing microencapsulated these bacterial cells, and we bring the subjects, 14 people, and what we did, they stay in a hotel in this center. They had a lunch, breakfast, dinner, and everything there for all the recent time of their studies, and we monitor their blood levels after feeding them these capsules. And these are what the results we got. And we published in British Journal of Nutrition. And this is a beautiful result. What it showed that we can reduce the LDL level by 8.9%, and we can reduce the ApoB level, total cholesterol, and triglyceride by 23%. So all these are very good if you look at the heart diseases, cardiac diseases. And what else we found, there is no change in anywhere, particularly fecal salt and fecal bile salts and microbial flora. So they were completely safe. We went further to see if we could feed for long term. So we did 9-week studies, And again, what we did this time, we did the capsule this time, in the capsule. And earlier studies were done in the microencapsulated yogurt. And what we found in this results that, yes, we can go as long as we want, and once we start feeding, cholesterol level goes down, and once we stop, cholesterol level will go up. And this one based on individual patients. So what we saw that it has a real effect. What also we saw is that the level of cholesterol and cardiac disease markers, they are going down as long as we are feeding and stop as when we stop it, and they're going back. So all these things resulted into a very good improved cardiovascular risk factor profile. And if you look at the risk profile in treated versus non-treated, you see that the risk profile decreases by 22%, which is a huge number in terms of not only cholesterol, but the overall risk profile. And similar fibrinogen, again, this is compared to the placebo versus treated, huge difference. So we had a pretty good results. What we also look at it, that each person, their bile salt, if you look at in the blood, their plasma, the bile salt level was down compared treated group versus non-treated group. So we see that deconjugation and the bile concentration is being affected by a single bacterial cells. And what else we found that it was affecting a pathways through a sterile absorption. And here there is another interesting result we found. This is a post hoc analysis. So what we found is the circulatory vitamin D is more bioavailable after getting these bacterial cells. And you can imagine the vitamin D is one of the major factors that you cannot take it through the capsules and through the pills. It is not as effective because they're not bioavailable as much as you can. And this is one bacteria increasing it and making this bioavailable. So we published that, and we started looking at all the mechanism. So what the mechanism we come up with is there is a bile salt hydrolase enzyme in the bacteria. When they put together in the artificial cells, they are able to break the bile salts into smaller molecules, deconjugated, and as a result you have a cholesterol catabolism, and as a result you have sterol excretion, and as a result you have a cholesterol decrease. So we come up with a complete mechanism in last 8 years, and we can say that there is a established pathways that is being affected by one single strain of a bacteria, and this pathways is through the bile salt hydrolysis. And this we published just recently last year. What else we analyzed? We analyzed to look at this, that how much dose actually you need for that particular bacteria. So this is the drug dose. You have statins, you need this many milligrams, and these are the plant sterols and all those natural products that people use it. And here is the single probiotic bacteria. You can see the efficacy of as good as drug. All you need is just 200 milligrams a day to achieve the cholesterol level decrease, similar like statins. So it was a very interesting result. So with this, we also try to understand the mechanism a bit more, and we see that as we go treatment per weeks, we see the level of a population of overall bacterial cells are changing. So what is happening is one bacteria is affecting bile salt pool, and as a result, many bacterial cells that are available in the GI tract, they are behaving differently and perhaps favorably. We analyzed that can we use this into Crohn and colitis diseases, because that is where also a lot of people are suffering, and we see that yes, there is a gene and there is a clear link between that bacterial cells and the genes associated with Crohn diseases, and we just published in PLAS One. With this dose size, we think it is as good as a drug, and what I want to see is, is that we have now well understood mechanism, a clinically proven multi-trial bacteria. It's patented, approved in US, Canada, and EU, and FDA approved. And I must say, this is the only probiotic in the world till today has a Health Canada or health claim approvals for cholesterol lowering. And none of that has available, and it can be used in capsules and in many places. With this, I want to say that human microbiome is a really undiscovered country, and we must look at it in a way so we can understand it, and we can understand the disease, use them for prevention and treatment of many diseases, and microbiome matters to the human health. With this, I would like to emphasize we are focusing on now more into neurological disorder and how the gut microbiota is affecting the brain, particularly diseases like Alzheimer, Parkinsonism, and amyloid, all those diseases. And we just published a paper in Cellular and Molecular Biology, and what we are looking at through a specific molecules, a specific bacteria can influence all these things. And it's going through the gut microbiota. So in other words, it looks like that human microbiome is playing more role than what we know, and it is being now really talked about, in particularly in medicine. In the beginning, people, when me and Dr. Chang were working in the beginning, nobody can actually think of that a bacteria can be used. So now people are talking about it, and everywhere it is microbiome is there, and we are looking at different ways. Essentially, we are actually covered with the microbiome. With this, I would like to switch to a different topic where we have used artificial cells, and that is in designing next generation of stent. So I will introduce you the problem of the stent, and I will introduce you why we need them, and I'll tell you what we think, and there are some limitations. So what we think is going to be next generation of stent is a gene delivering stent. It will be a game changer. That's what we think. So the problem, cardiovascular disease, again 1 in 4 in all health, death 17 million worldwide, and blockage of coronary arteries, heart attack, again something like lipid and cholesterol and high blood pressure and heart attack. So we have over 1.1 million stents used today in US, and is costing around 12 to 30,000 per patient. And what we have reached is really remarkable. It's become a care of a standard. People have used a stent and are using a stent, and they're very healthy for a long time. So really they're very good. So what you're looking at is, even though they're good, but they have a 2 biggest limitation. One is in-stent restenosis, and other is in-stent thrombosis. And these 2 are still the problems. And drug-eluting stents are the best one, but they react to the consequences, and particularly endothelial damages. So we thought maybe there is a room to overcome these drug-eluting stent problems. And if we do that, we could really meet a good clinical need, and particularly through re-endothelialization of the stents, of the blood vessels. So the solution, what we think is actually you need to have a wound repair at the place. So we come up with an idea that can we target this with a genes, and this is where artificial cells kicks in. So what we had a gene, and we wanted to get into to the area on the stent. So what we did, we have a modified baculoviruses, genetically modified, and then we put into the artificial cells, and we coated them on the stent itself. The idea is to have a matrix that will have all the materials inside, and then you coat them, and essentially it can be used in many places, perhaps in the stent, orthopedic, soft tissue, skin, and all places. So these are the main technology that we develop is the baculovirus, and baculovirus put together, coated in a vessels, and then once they go inside, they will release their mRNA and they will actually do the repairs. So local targeted delivery using artificial cells was the key behind this technology. So let's look at the earlier people have tried about the viral genes. They have used adenoviruses. If you look at them, they have a lot of limitation, particularly infections, and they induce immune response. Keeping this in mind, we come up with baculoviruses. So the baculoviruses are insect viruses. We all have it. We have so many, and they are good about it because they do not replicate in mammalian cells. So if they come to us, they do their job and they die. They do not elicit any immune response, at least only one report is there, and they not integrate with the human viral DNA. And they are classified as safe by FDA, as safe as you can eat them. They are used in manufacturing vaccine, influenza vaccine, and all those vaccines. So they are used and they are everywhere in the natures. They are perhaps more than microbiomes in the nature. So to overcome these limitations, what we did, we put these engineered them, baculoviruses, to produce protein of marker and repairs of endothelial cells, and we put them inside the microcapsules, artificial cells, and we coated them in the stent. The idea is behind is that if you deploy them, these baculoviruses, they will excrete out the proteins locally after infecting the cells, and these cells, they will help them, and they become biodegradable, and they will die after a certain period of time, and as a result, you have a functional growth release. So, excuse me. So this is a damaged endothelial cells. This is where the problem is. You have all endothelial cells, you deploy the stent, and they come back, and as a result, you have a stent failure. What our idea was to, if we use artificial cells to make a surface in a way that they will actually come into, and they will re-endothelize the whole thing. So it's a local repair. We did this, and we tested in an animal model, and particularly dog, and the pigs. This data I'm showing you is from the dog. So we took damaged their arteries, and we implanted our stent in a way, and we saw the repair of the pretty good. And we see is real reduce in intimal hypoxia. And therefore, we concluded, and this is based on the protein that they are excreting locally, the cells, we concluded that yes, this can be used and deployed in human, and we published in Nature Scientific Report. So where we are is we can do a coating, we can keep at the room temperature for these baculoviruses for up to 3 years. We can dry them, even though if they have artificial cells on the table. We can store them in the room temperature, and we can maintain the viability. So we are ready to go for human. So this is where I come up to developing an industry, setting up a company, and this is where this company, from there the company is taking over and doing this research. So what is the advantage of this stent is it has, unlike other cardiac stent, it's fixed the problem. It's not just only make a repair, but it's fixed the problem through re-endothelizations. We think we have a better clinical outcome because of reduced clinical complications, reduced need for the dual antiplatelet therapies, reduced need for future cost bypass surgeries, and increased longevity of deployed stent. Baculoviruses are inexpensive to produce, just $4 or $5 to have one stent, including production of artificial cells altogether, and we think it will give a big benefit to patient, clinicians, and the government and insurance because of the reduced cost. So in conclusion, what I want to conclude is microbiome and stent are very important for human health. But most important is artificial cells. And this artificial cells, as you all see, has been shown to be effective in many areas, and it is making a strong impact in both microbiome and stent. And it's really is up to us to use this game-changing science to various humanity and to treat and understand diseases and I'm sure this will go beyond that. With this, we are working in artificial cells, and we are encircling, and I try to coin a term based on Dr. Chang's theory of encircle everything from the beginning itself. And we are encircling for technologies for human health, and we think that we will make an impact through the artificial cells. With this, I would like to thank Canadian Institute of Health Research, NSERC, FRSQ, and Ministry of Economic Development, and all these people throughout many 25 years of work. So thank you all. And I would like to show you our visitors, Montreal look like that in a couple of months. But you're always welcome. We have a very warm city, and the warmest center we have is Artificial Organ Research Center and the McGill and Montreal. You're always welcome. Thank you.